as long as I can remember, I've loved going to the beach. Many times my brother and I just walked the shore, looking for shells, skipping stones across the water surface. But I've started thinking about things I've read in the news about how our ocean, and more specifically our tropical reefs, are in danger of being changed forever. I never really thought of them as living things, but they are. I wondered how it happened and if anything was being done to assess and manage threats to Guam's reefs. I decided to visit the marine lab at the University of Guam to find out.
Hi, I'm Elijah.
Hello, Elijah. Welcome to the Marine Lab.
Thank you for taking the time out to talk to me, Dr. Shills.
So what specifically is happening in Guam?
Well, Elijah, it's not just Guam. In fact, globally, reefs are in danger. In Guam, we've seen a lot of change taking place on our reefs. In recent years, since 2012, actually, a certain type of alga, the angel hair alga, came out of nowhere. It had gone unnoticed for at least 3 generations, that fishermen, beachgoers, nobody had seen it. Even marine biologists had never documented in the field. And suddenly it occurred on our reef flats and covered about 1/4 of our reef flats in its totality, basically. So shading other reef organisms and completely covering reef flats that are composed of other species otherwise. Another kind of threat that has been documented for Guam for the first time in the 1970s is, for example, a sponge, Terpios, that overgrows corals and other organisms on the reef. And since it's for the first time been recorded in Guam, the phenomenon basically of these outbreaks for the first time documented in Guam, it's now been seen elsewhere in the Indo-Pacific where it's causing problems. Then we also have bleaching events, coral bleaching events that have become more frequent in Guam and also more severe. So basically increased temperatures, ocean temperatures over a large period of time with increased irradiance have caused corals to become sick. That can be seen as like the corals become more pale, they bleach, and shortly after generally these coral colonies die. Other types of changes that we've been documenting is more coral diseases on our reefs, and that's often linked with pollution. We also have crown of thorns sea star outbreaks in Guam. They were first described from Guam, and they are now once again causing problems elsewhere in the world. But basically, we suddenly have outbreaks of these sea stars on our reefs, and they devour healthy corals and basically eat up the living tissue of these corals over large extents of reef. We have also intense fishing practices in Guam. And those intense fishing practices are generally targeted to specific groups of fish, which can bring the ecosystem out of balance. Then we also have sedimentation, and sedimentation is generally linked to periods with heavy rainfall, and in areas where there are, for example, badlands, sediments accumulate in the runoff and ultimately smother the reef organisms and have particularly negative effects for organisms that depend on photosynthesis on the reef because the light that reaches these organisms diminishes. And then we also have like a more localized disturbance, vessel groundings, for example, that is basically a physical impact on a reef. And this is generally more localized, but in recent years we've experienced quite a few vessel groundings around Guam.
Okay, I understand. So do we know when and where this is happening? Are we tracking this?
Yes, recording these disturbances is important for reef management, particularly to evaluate what the long-lasting impact of these disturbances is. So at the Marine Lab, we're involved in a monitoring project in collaboration with the Department of Defense, and that consists of 2 entities on island, Naval Base Guam, and then the Naval Support Activity Andersen Air Force Base. Our monitoring effort focuses on 2 areas in Guam. One of them is Apra Harbor, and the second one is the North Shore up north next to Andersen Air Force Base. So Apra Harbor consists of sheltered reef communities and is truly unique in the world in the composition of the reefs that we find there. This is also an area that has been altered by man historically, and particularly since the Second World War. For example, when the glass breakwater was built in Apra Harbor. Afterwards, a lot of in-water construction activities took place there, as well as dredging activities. So these are the reef communities next to Naval Base Guam that we studied. And then the other study area, the North Shore, next to Andersen Air Force Base, is an area that is hydrodynamically very different. So there's a lot of more flushing that goes on there. The reefs are a lot more exposed to wave action, and in some studies it's been proposed that those reefs act as a refuge for some coral species that suffer from coral bleaching elsewhere around the island. So the reef communities up north represent the fringing reef communities around the majority of the island.
So this coral reef monitoring, is it just for corals?
So no, we don't only look at corals, we also look at other organisms, and in fact we're a team of 4 researchers at the Marine Lab. So we have Peter Hauk, whose work focuses on the coral communities. We have Jason Biggs, who studies the mobile invertebrates, and this is a group that typically is not being included in monitoring studies simply because they're too complex and too difficult to study. And we have Terry Donaldson, who studies the fish populations. And then I've been involved in the studies of the benthic organisms. So these are the organisms that are attached to the reef substrate, and of which a lot of them make up actually the reef, build the reef. That's at the macroscopic level, so these are the organisms that we can see with the naked eye. Then I've also been involved in the study of the microbial organisms or microbial communities associated with corals, and that's a newer approach to reef monitoring, ultimately with the goal to bring the cost of reef monitoring down and to be very accurate in detecting and anticipating changes of our reefs. And then I've also been involved in the water quality monitoring, and for which we have installed oceanographic instruments at some of our sites in both study areas.
So what have you discovered? What has your research told you?
So in this first phase of the monitoring effort, everything is focused on baseline. So we've established baseline data for all the groups of organisms that we studied, and also for the water quality parameters. Besides that, we've characterized the different reef communities of the North Shore and Apra Harbor. And that has guided us into a direction that we now know how to analyze those data efficiently in order to derive information about reef health. In general, we found that we have to use a different strategy for the northern communities, the North Shore and Apra Harbor. But we've also found that we can reduce our survey efforts severely by focusing on a few groups that are ecologically informative. So these are groups that are important for reef health, ecologically important for reef health, that occur in a large amount of biomass on the reef, so that they are, from a statistical perspective, informative. And those 4 groups of organisms are turf algae, hard corals, coralline algae, and then calcified green algae. And of those 4 groups, there are 2 groups, the hard corals and the coralline algae, that actively build reefs.
So what is your process of gathering data?
The process of gathering data, indeed, I think the most interesting component might be the fieldwork associated with collecting these data. So, as I previously mentioned, we're a team of 4 faculty members and some students that all work together on the same sites. And we have like 10 sites in Apra Harbor, 10 sites up north. For the fieldwork, we all gather on a boat and do dives together. So we jump in the water, dive down, establish transect lines, and along those lines we collect our data. This differs between the different types of groups. For me, studying the benthos, so basically all organisms attached to the reef, we calculate the cover of each of the organisms on a reef. We use quadrats for that and digital images to calculate those percentage covers. For the coral surveys, they actually measure each of the colonies. they find in these quadrats, so that they can come up with a size distribution of coral colonies for each of the different sites. For the fish surveys, they swim along the transects and they count all the fish that they encounter and identify them to species level. And the same happens also for the mobile invertebrates. Clearly the mobile invertebrates, they then focus on the substrate and they look in small cracks and crannies on the reefs, on the reef to establish basically the total account of the different species and the abundance of some of these species on a reef. We also install at these sites water quality sensors, and these water quality sensors, they collect data continuously. So we deploy them during one of the dives, and then when we revisit a site, we bring up the instruments, download the data, and redeploy them. So we have a continuous time series of data, which is unique for Guam thus far. And then finally, we had also a pilot study on the microbial organisms. And here, for this study, we looked at the microbial organisms that live in coral skeleton. Some of these organisms are beneficial to the coral host. So during times of stress, they can feed the coral host sugars so that they can endure those periods of stress better. Some of these are detrimental to the coral host. They bore into the coral skeleton, make it more brittle, and so the coral colonies would disintegrate, like if we have extreme storm events or whatever. So we study the organisms in those communities, those of tiny organisms. We can hardly see them with a microscope or not even identify them with a microscope, but we sample them and we break off a small piece of the coral skeleton and try to characterize the communities, the microbial communities in this coral skeleton using modern approaches to DNA sequencing. And what we found is indeed we find similar patterns as whatever we observe the macroscopic organisms, the one we can see on the reef, that is also mimicked by those microscopic communities. And that opens interesting avenues for future research, actually, because we can employ such methods also maybe to other reef organisms, that in the future we can take water samples to characterize, for example, the species diversity of fish on a reef, a very accurate identification of all the species that occur in a certain area, even fish species that are very wary of divers or people in the water altogether, particularly linked to the intense fishing pressure in Guam.
So what can people like me, what can I do to help?
Yes, clearly, taking care of the environment is a priority, and it can start with each one of us. There are a few simple steps by which each one of us can contribute to a healthier environment. So first of all, littering is definitely out of the picture and should be avoided at all costs. So reusable containers, reusable water bottles are a must. Also, the release of non-native animals and plants should be avoided at all costs, and particularly in the case of, if we discuss the marine environment, like non-native plants and animals from reef aquaria should not be released into the wild. Then also participating in outdoor activities like snorkeling is definitely encouraged, but try not to disturb reef life. Do not step on corals or do not take any animals or plants out of the water, right? Another thing we can do is eat locally produced food, but eat wisely. When possible, opt for pelagic fish like wahoo, mahi-mahi, or yellowfin tuna instead of reef fish, as these pelagics have fast growth rates and healthy fish stocks around Guam. Then another thing we can do is report violations of illegal reef harvesting or fishing in protected areas to law enforcement agencies. And also report wildfires and do not participate in off-roading activities, as they cause erosion and increased sedimentation on our reefs. You can also participate in tree planting events, which aim to restore native forests on island, which prevent, once again, erosion. And then lastly, talk to your friends about the beauty and amazing complexity of tropical reefs. This way, you can pass on your knowledge and increase the number of people interested and committed to protecting our marine environment.
So although the situation with our reefs is something to be concerned about, there is hope that if we all do our part to be environmentally conscious, we can maintain our lush and healthy reefs for generations to enjoy.